Yeah, thank you very much for the kind introduction, and I'm still totally struck by the speech, in fact by all the speeches that we've had so far, and I think now my task can only be to piece them together in a slightly different way, because all the important things have been said one way or the other. Luckily, I have also prepared a small scientific lecture, so that at least I'm on slightly safer ground afterwards. Yeah. So if I appear a little bit insecure at the beginning, then perhaps I will come into safer waters later on as we go down. Now, I was extremely happy with your presentation just now, Hashim, with two exceptions. The first one was that I think my role was vastly exaggerated. Yeah. The program, Molecular Life Sciences, or MoLife, was a community effort of the entire life science community, and without the amazing people, the colleagues, and also in fact the fantastic students that we had. I mean, you know all the people in your cohort, and but there were many other cohorts, and they were all very, very good people who went on to do amazing things. Yeah. And I would say exactly as, actually Predrag said this before, that in the true Jakob spirit, yeah, I think all of us hope that eventually the university will come back to providing master programs like this that allow people like you to do the unbelievable, amazing, fantastic, beautiful things that you do. Yeah, I don't think I've run out of adjectives here. Yeah. Just maybe that I've never seen anything like this. I've never seen anything like what you have recently achieved. And this brings me to my second point that I disagree with, because I thought that we were on first name terms actually. And now it's me. Yeah, and now it's me to to say that we will be very lucky, very fortunate in the future to be associated with you. Yeah. So rather than you being associated with Jakobs will be proud to be associated with you and your future enterprise. Now that brings me a little bit to the things that, in fact, our dean has pointed out, Dean Kappas, just a few minutes ago. And this is that some of the things that you have initiated, or that you have reminded us of, that you have started out, are things that we must, I wouldn't necessarily say refocus on. We've always been very interested in these things as a university, but they're things that we ought to think about perhaps a little bit more. The first thing is definitely training master students. I think this is something everybody wants. Yeah. The second thing, very, very, very important, as the dean pointed out, science communication. Yeah. Now, science communication is not easy. Yeah. I don't know how you do this. Yeah. I have not the slightest idea how you manage, but I feel that my talks to a general audience are usually met with sympathy, but also not terribly often with understanding. Yeah. We'll see how this goes in a couple of minutes. Yeah. But this is something that, and I really do agree with the dean, and I agree also with Alexander Lerche, that this is something that is crucial. We are spending public money on our science, and we need to explain it. And we need to explain it not just in terms of what is it useful for. Yeah? So what drug, what machine, yeah? Which faster car, yeah? What moon rocket, what genetic engineering of embryos or whatever else. Yeah? But also, I want to, since I get to speak, yeah, I want to make a point for basic research, yeah? For curiosity-driven research that just asks, why is it like this? Why is that so? Yeah? Because, to my mind, this is something deeply, intrinsically human. Yeah? The endeavor, the enterprise, the aim to just understand what's happening around us. Yeah? And to get this across, this basic wonder, yeah, of humanity, yeah, when faced with cosmos. I'm not a star person there, but some people are. Yeah? When you're faced with stars and planets, when you're faced with tiny little crawly objects, yeah, Also, how does this work? What does it do? Yeah? And these things, I mean, you know this very well. In the end, this curiosity has led to all of medicine, medical research and drug research and all of these things. And this is the basis. And I'm so glad that we can do this here at Jacobs. Yeah? But we have to redouble our efforts here at Jacobs, exactly like the Dean said. We have to redouble our efforts here to teach people to communicate, to give talks, to present, to go out. Yeah? And my dream, I know you have a lot of dreams, actually a lot of them you have made come true. Yeah? One of my dreams would be to help the students here, including, of course, PhD students who are thinking, you know, what should I do with my life? Yeah? To encourage them to go out and communicate about what they do. Yeah? And maybe this is something where you can help us understand how this works and do this a little bit better. Yeah? So this is, and these are very important stimuli, very important things that that you can teach us. Yeah? We have taught you what we know. Now is the time for you to teach us what you know. Before I get cut off, you're not looking at your watch, but no. Okay. Anyway,
the alarm set.
I'm going to briefly, very briefly, explain to perhaps to people, to everybody, what are we going to do. Thank you very much for your donation. It is deeply appreciated, and it is deeply appreciated because it allows us to generate, to purchase, to put into place a piece of equipment that we need urgently. Yeah? And I'll explain to you very briefly why that is. Brief lecture on tumor immunology. In the last few years, people have come up with a revolution of the therapy of cancer. And I know this is an unpleasant subject, but you know, many people know people who have suffered at some point, and it's deeply, it's also for me, because of cases in my family, it's a deeply personal issue. Cancer cells, it's relatively easy, are cells that suddenly stop behaving, they grow, they keep dividing, and they cannot be hindered anymore. And so as such, they can cause serious damage in organisms. In the old days, one has taken drugs, one still does this, one takes drugs to kill these cells, one uses surgery, one uses X-rays, yeah, to destroy tumors, but people remembered more recently that our immune system contains cells, the job of whom is to kill other cells. For example, cells that are infected by viruses, or cells that have aberrant conditions that grow and divide and that become tumor cells. And here's one example. You have a little immune cell on the left, the so-called T cell, and you can see that it attacks another cell, and the other cell then eventually dies. So we can do this. Our immune system can do this. Our immune system could kill cancer. Why does it not do this? Oh yeah, before and after. Why does it not do this? Oh yeah, first, perhaps very briefly, I don't have a pointer here, but it's not so terribly important. The main star of the show is this red object that we call in the lab an MHC class 1 protein. And this is very important because in the tumor cell, this ugly cell at the bottom, it grabs some little fragments of proteins, you don't even need to know what a protein is, carries them to the cell surface, and the T cell, can you see this at the top? The T cell can look at that and it can see, oh, this is an infected cell, this is a tumor cell, this is a cell that we should eliminate. That red protein is in red because it is the most important one. It's the main player in the show that is the research in my laboratory. No. Why does this not always work? Why don't T cells always kill tumors? This is very simple, because tumors, they do everything that they can to prevent these T cells from killing them. they secrete some substances like hormones that make the T cells unhappy and depressed. They try to hinder the T cells from working. So for a very long time, people thought that immune system cannot kill tumors because of this so-called tumor immune escape. Now, there is a new way in which this can be prevented. One can actually take, from the very left, one can take the T cells from the patients. One can grow them up in the test tube, in the culture lab. One can activate them. And then, on the very right, one can put them back into the patient, where they go to finish off the tumor. This is the future of tumor therapy, and I think you wrote about this, or you showed a video about this already a very long time ago. Yeah? For you it's not new, but this is what is happening at the moment. The slight problem, if you want to find the tumor-specific cells, is that all the T cells look the same, but only very few of them are the ones that we want. All the T cells are tiny, small, round little objects in the blood, and nobody knows how to find the right ones among them. And this is why scientists, they use a reagent called an MHC tetramer, and that is seen here on the right. It's a pretty complicated object. All these colored parts, they belong to it, and you can see again our red protein. This time it's not in the cell, but we made it in a lab as part of a reagent to detect the T cell. The T cell is now seen at the bottom, to detect the T cell. Once you know how this goes, once you've detected something, you can pull it out. You can isolate it, you can grow it up, and you can put it back into the patient. So the thing that we work on at the moment a lot are these things called tetramers, or MHC tetramers, because we use them, people can use them to identify and isolate T cells. That's for the super experts, this is what it looks like. I'm very happy to explain it in detail in the discussion if you want. There'll also be a small quiz. So we will, we will see how it goes. I stole this picture from my PhD student Raghavendra that shows it actually what the preparation of these tetramers involves. In fact, this is not exactly the preparation of the tetramers, but it's the preparation of the proteins for something, for determining the structure of them to see that everything is fine with them. And you can see that step number 3, step number 4, step number 5, they involve something called chromatography. So you could say, without chromatography, we cannot make that reagent. Make sense? Oh, just because I stole Raghavendra's picture, I wanted to show one of his data slides. This is the structure of our favorite molecule at the bottom, and at the top it's bound to a little part of the T cell. This is something that was generated in our lab. I'm very proud of it, and thank you, Raghavendra. All these things that are called chromatography, they are conducted with this kind of instrument, and this is what I'm not naming any companies here, but this is the kind of machine that we intend to use the money for. This is the idea. And this is something that is very hard to finance otherwise, because it's an expensive instrument. Yeah. And it is something that is at the key of many of our approaches in the lab. And it is something that, I think this is what Britta pointed out before, something that we can eminently share, yeah, with other groups in the building. So that this means that your donation will be useful not just for us, it will be useful for the entire life sciences community, because so many things interdigitate and overlap here in terms of science. And this is, I think, it's exactly what Britta said before. This is something that makes Jacobs an especially exciting place for the life sciences, is the sharing and the interaction and the connection. We're not the MIT, not yet. Yeah? We're not Caltech, not yet. Yeah? Give me a few years. Yeah? But we do something that people there often don't do enough. We talk to each other. Yeah? And as long as we keep doing so, we just keep coming back to this theme of communication. Yeah? As long as we talk to each other, yeah, it cannot go wrong. It will eventually work. So I would like to say, okay, this is just. some of the things that we recently did, patents and licenses, but since time has gone on so far, I just wanted to say from all of us, thank you very much to you, and thanks very much to all of you for listening to this long presentation.